welcome to The Late Show. This evening's topic is going to be signature in the cell. And as I've been preparing to interview someone far smarter than me tonight, I've just reverted to scriptures that we all know well, that God's eternal power and divine nature have been clearly seen, being understood from what has been made. And that's in Romans 1, and Hebrews 1 is equally wonderful, talking about the Lord Jesus being the radiance of God's glory, the exact representation of his being, sustaining all things by his powerful word. So keep that in mind as we go into the detail and the intricacies of the cell. And we shall sort of bring your questions in later by email and text. But please hold them, because we've got a lot to go through with our special guest this evening, Dr. Stephen Meyer from Seattle, Washington. Stephen, welcome to the show by Skype.
Nice to be with you again, Tim.
It's great to see you. Thoroughly enjoyed the discussion we had at the end of last year on Darwin's dilemma, or Darwin's doubts, and really excited to discuss this subject. Now, you've written a book called Signature in the Cell. I was very interested to see on the copy, on the front of the book, that it won the Times Literary Supplement Book of the Year prize. How did you manage that?
Well, it was one of a number of books commended by the Times Literary Supplement. It wasn't the only one, but it was recommended for that designation by Thomas Nagel, a very prominent philosopher of science at New York University, who himself happens to be religiously agnostic and has tended even towards atheism in his position. But he was fascinated with the discussion of the problem of the origin of the first life, which is a vexing problem for people working in evolutionary biology. And he liked my discussion of the problem, and has defended me at some pains to his own reputation. I'm afraid he's been savaged by some of the really staunch advocates of Darwinian materialism.
Because in a nutshell, your book is saying that there is a signature in the cell, so he must have got into hot water just by acknowledging that title.
Well, right. The signature in the cell title is in a way a metaphor for the ability to infer design. There's something in the cell which is, I argue, a signature or a distinctive hallmark of intelligence, and that distinctive hallmark, I argue, is the information that is stored in the DNA and RNA and these other large biomolecules that are inside the cell that are running the show. Information, as we'll discuss some more, I'm sure, runs the show inside life. And so I argue in Signature in the Cell that we're actually looking at evidence of the action of a prior designing intelligence. Nagel himself is reluctant to go that far in his own understanding. He has come out with his own book called Mind and Cosmos, subtitle, How the Neo-Darwinian Materialist Understanding of Reality is Almost Certainly False. That's his. He published that a year ago, well, actually fall of 2012, with Oxford Press. But Nagel himself does not advocate intelligent design, but he likes many of the critiques that those of us in the ID movement have made of these materialistic theories of origins.
Now, one of the things Paul says to the Corinthian church is he's not peddling a message for profit. So, you know, to peddle something for profit, you seek to be popular, as it were. But your position, you know, on intelligent design within your peer group, let's say, and within the scientific community isn't necessarily a popular message.
Not necessarily popular. That's nicely understated, right? It's been very controversial. And I would say it's a very upsetting and disturbing message to a lot of people in the sciences and in the philosophy and philosophy of science fields that are concerned with these questions. And yet at the same time, I would say there is a growing community of scientists and scholars who are intrigued with this idea and who see the force of the argument for it. So I think it's a very exciting and dynamic debate to be part of, but almost no one is neutral about it. And you see that when I think we were talking last time when we talked about the release of my book last summer, Darwin's Doubt, and the the bimodal distribution of reviews on internet websites like Amazon.com and Barnes and Noble. It was getting either 5s or 1s, and so people were they either really liked it or they were really, really troubled and upset by it, and you could see that in people's reaction.
Okay, now one of the challenges we've got tonight is to sort of somehow break beyond the scientific community without losing sight of them and sort of demystify the subject a little, because for many people watching, for many of you, our viewers, you know, some of the terminology can appear gobbledygook, and it's just a challenge to somehow break it down to a sort of language which is comprehensible. And so that's what we'll try and do this evening, if possible, Stephen.
Well, let's work hard at that. We've got some animation that'll help, and I've got a couple visual aids as well that I use when I'm giving talks on this. So we may break from ordinary broadcast protocol and use some visual aids.
So just talking about the animation before we put it up, it's absolutely amazing animation by a chap called, I've written his name down, Joseph Condelis. Is that correctly pronounced?
Right, Joseph Condelis. Yeah,
absolutely excellent. Really fantastic. And I think that what we'll do is we'll show this. It's a few minutes, a clip called "Journey into the Cell," which highlights some of the points that we're going to be discussing. So we'll show this one. We don't want to show the second clip, if that's okay, control room.
Tim, can I give just a little bit of
an intro? Please do.
We'll set some context for your audience.
Great.
The book we're discussing, Signature in the Cell, is addressing a very fundamental question in biology, which is the question of the origin of the very first life, not the origin of complex animals or the origin of human beings, but the origin of the very first living cells. And many people don't realize this, but this is a question that Darwin never himself addressed. He wrote in a letter or two, he had some speculations that he expressed in some private letters, but he didn't address the question of the origin of the first life in his book, The Origin of Species. He was attempting to explain in The Origin of Species the origin of new forms of life, always from simpler pre-existing forms, but he presupposed that there was a first living cell to get the whole process of evolutionary biology going. And so this book addresses that even more fundamental question, which is, how did we get life from non-life? And in order to answer that question, you have to look carefully at what even the simplest cell is like on the inside. And this animation shows what's going on inside even a very simple one-celled organism.
Wonderful. Okay, so we're going to journey into the cell to see the signature in the cell, and we shall come back to you shortly.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Welcome back. We are in discussion with Dr. Stephen Meyer on a signature in the cell. And Stephen, at the beginning of that clip, it's had in bold writing, where does the information come from? So I suppose that's the overriding question when you see the complexity of just that animation and all the elements of it.
That is the crucial question. And as we were saying right before you played the animation, this was the question of the origin of the first life was not a question that Darwin himself addressed in 1859. And it was a question that didn't seem really to trouble other biologists and evolutionary thinkers in that early period of the late 19th century. And one of the reasons for that is that they assumed that the cell was an extremely simple entity made of a simple chemical substance called protoplasm. And one of Darwin's contemporaries, Thomas Henry Huxley, said that the cell is a simple homogeneous globule of undifferentiated protoplasm. It's basically some kind of goo that or jello that would form, and they envisioned that it might have easily formed with a few simple chemical reactions. What we now know is, this animation, as intricate as it is in some ways still a cartoon version of just how complicated the inner workings of information processing systems are within cells. But since the 1950s and 60s, post Watson and Crick and the molecular biological revolution, scientists have determined that what makes cells function is precisely this kind of complex information processing system, and so if you want to explain the origin of life, you have to explain its nature. You have to explain what life is actually like, what we see before us. How did the systems that we now study arise? And so that puts front and center the question of the origin of the information encoded in that DNA molecule, which is directing the construction of the proteins and protein machines that are necessary to keep all life, cellular life and animal life, plant life, going. So that's the key question. Where does the information come from in the first place?
Now, you know, we can look at Darwin and say, well, Darwin sort of missed that off his list of questions. But, you know, the folks that still follow, you know, neo-Darwinianism or whatever, you know, how do they answer it today? You know, do they suggest that somehow life is inevitable and these processes are inevitable, that, you know, somehow automated, you know, without any guiding force?
Well, that's a great question. Let me make a quick distinction. There are two branches of evolutionary theory. There is biological evolution, and Darwinism and neo-Darwinism are examples of theories that are biological in nature. They're attempting to explain the origin of new forms of life from simpler pre-existing forms of life. But then there's another branch of evolutionary theory called chemical evolutionary theory, and that branch of evolutionary theory is attempting to explain the origin of the first life from simpler pre-existing chemicals. And the subject of signature in the cell, my first book, was this question of the origin of life. And so the question you're asking can be, if I can just slightly
Sure.
amend it, it's how do chemical evolutionary theorists account for the origin of this, the information necessary to build the first life? And that has indeed been a very vexing and difficult question. They've had three basic different approaches. One is to invoke just brute chance, lots of time, lots of random interactions of molecules. That's one approach. Another approach is the approach that you just alluded to, that sometimes goes under the heading of necessity or self-organization, that the molecules out of which the DNA are made have some inherent properties of attraction that cause them to align, to cause the parts of the DNA to align into these informational arrangements. And then the third idea is somehow combines chance and these forces of chemical necessity. Okay. And in the book, I look at all three of those different approaches and show, for various reasons, both experimental and empirical and theoretical, that each of those three approaches fail and have failed to account for the origin of the information necessary to build those information-rich DNA molecules that are so critical to life's function.
Okay, could I have a crack at chance in this question? That the you know, one of the arguments that goes back to my school days is that, you know, if it's an infinitesimally small chance, you could have an infinite number of universes. So, and I've heard you say that it's like the chance or the odds are like finding a needle in a haystack the size of the universe. So why not have, you know, an infinite number of haystack-sized universes?
Well, this is gets to the multiverse idea, yes. And that takes us really out of biology into cosmology.
Sure.
A short answer on what's wrong with the multiverse is, or rather, the way I'd frame that is, there are reasons to prefer a theistic design hypothesis as a cosmological, as an interpretation or as an explanation for the fine-tuning both of the laws and constants of physics, which is something we haven't talked about, and of all of the conditions that would be necessary to get life going, which is what we are talking about. There are reasons to prefer a theistic design hypothesis over and against the multiverse. One very simple reason to prefer theistic design is that both of the cosmological models that justify positing a multiverse, billions and billions and billions of other universes, is that both of the cosmological models that justify that postulation themselves require fine-tuning to get a universe like ours. So that one of those models is called the inflationary cosmology, and to get universes like ours, the advocates of the inflationary cosmology acknowledge that you have to finely tune something called the inflaton shutoff energy. And string theory is another cosmological model that might give you a multiverse, but the string theoretic landscape, as it's called, must also be finely tuned to give you a universe like ours. So even in those really speculative models of the universe that involve postulating billions and billions of other universes, there still has to be a mechanism for generating new universes, and those mechanisms themselves have to be very finely tuned, which just pushes the design question back one generation.
Got it.
So I don't think the multiverse solves the problem.
Okay. What I'm doing is complicating an already quite complex subject. So we'll get back to the cell.
If we can just confine the discussion to the biological context,
sure.
People trying to understand the origin of life as a matter of the random interaction of molecules within this universe,
yeah,
have pretty much given up on, in fact, I would say have entirely given up on models that rely on pure chance, for the simple reason that the odds of getting all of those elements, chemical subunits that make up the information-rich messages to align properly so that they actually contain information for building proteins, the odds are so very small that it's overwhelmingly more likely that random processes will fail than that they will succeed in the entire history of the universe. If we take into account all the opportunities there are for building molecules in that random way. So there really aren't any serious origin of life researchers who are proposing pure chance as an explanation anymore.
Now, Stephen, yeah, I've got that now. Now, we're showing on the screen the animation again, and at this point, I think the RNA is going through the nucleus, the filter in the nucleus. It just seems that every stage of this animation, let's go, or this process, it just looks extremely complex and orchestrated.
It's complex, but not complex in the sense of being merely random. It's complex in the sense of integrated complexity, the kind of complexity that engineers produce, where each part is placed in careful relationship to other parts so that the ensemble of elements and parts performs a discernible function. And this is what we have at every level in biology: specificity of arrangement and complexity. And it's really impressive. And it was a kind of, well, very surprising discovery in the 1950s and 60s during this period of time historians of science call this molecular biological revolution. In the 30s and 20s, people were still hoping that the secret of life would turn out to be something very simple. People hoped that proteins would stack neatly on each other. They would have simple structures like piles of cigars in a little pyramid shape, or stacks of hat boxes, or something that would be very simple and repetitive, like maybe something like a crystal of salt. But instead, both proteins and DNA turned out to be these incredibly complicated. Protein has a complex three-dimensional shape. DNA has this complex informational character, and these two molecules are key to everything else that is going on inside the cell, everything else that gets built. And
it's part of the regeneration process, isn't it? Because, you know, if there wasn't a regeneration of cells, we wouldn't last very long, would we?
Well, exactly right. And just copying DNA, a process called DNA replication, is a fascinating process by itself. And you have this problem as well, that in addition to the information stored in these molecules, there are the two types of molecules: the DNA, which is sometimes referred to whimsically as the tape, and the proteins, which are the shapes. So the tapes make the shapes. The information in DNA is directing the manufacture of these intricate three-dimensional structures, which perform functions in virtue of the very specific shapes they have that allow them to catalyze other reactions, or build various machines and structures, or process the information and turn on DNA. That's another huge problem. You need DNA to build the proteins, but you need the proteins to process the information on the DNA. So you have this really intractable chicken and egg problem that is it's something else I discussed in my book. It's one of the real problems for all forms of chemical evolutionary theory.
One thing that comes across watching the animation is that it's not just an engineering process, let's say, of stamping out saucepans or stamping out shapes. It's as though there is just this intricate, synchronized process going on, which somehow needs a guiding force. It doesn't look to me as though you could that it's inevitable or automatic.
Well, it is automated if you presuppose the basic laws of physics and chemistry holding atoms and molecules together. But the big question is the origin of the very specific arrangements of those individual DNA and protein molecules, and then the ensemble of molecules that form these complex systems. A really good analogy, I think, is to what is commonly used now in manufacturing, human manufacturing, whether it's automobiles or airplanes, or I talked to recently a guy I know who's involved in manufacturing garage doors. And lots of manufacturing plants now use a technology called CAD/CAM, computer-assisted design and manufacture. And what the engineers do is write code that is then translated into a machine language that can direct a manufacturing apparatus to produce some mechanical part. So here in Seattle, we have the famous Boeing plant. At Boeing, the engineers are writing code to direct, for example, the placement of rivets on an airplane wing. And so you have information directing the construction of mechanical parts, and that's exactly what is going on inside cells. And so to explain the origin of even a simple one-celled form of life, you have to explain the origin of the information that's directing that process, as well as the information that was necessary to build the protein parts that make that process possible.
Yeah, what really strikes me, looking at it, listening to you, is how people can come to a conclusion other than that it was designed. How can they look at it and say that it was not designed?
Well, as I mentioned, there have been these different approaches: chance, self-organization, trying to combine chemical necessity with chance.
Yeah.
And in Signature in the Cell, I critique each of those approaches in some detail. But I think it might be helpful to explain the positive logic of the inference to intelligent design based on the evidence that we see in systems like this. We know from experience, our uniform and repeated experience, which is the basis of all scientific reasoning, that it takes a programmer, for example, to build a computer program. Bill Gates, Richard Dawkins, others have acknowledged that DNA is very much like a machine code or a computer program. And we know from experience that it takes a programmer to make a program. More generally, we know that from experience that whenever we see information, especially in a digital or alphabetic form, and we trace it back to its source, whether we're talking about a hieroglyphic inscription or a section of computer code or a paragraph in a book or even information embedded in a radio signal, when we see information and trace it back to its source, we always come to a mind, to an intelligent agent, not an undirected material process. So the presence of information at the foundation of life, performing those functions that we just saw in the animation, is a very powerful indicator, I argue, of the activity of a prior designing intelligence. Information requires intelligence. That's what we know from observing the world around us. And so when we encounter information, when we're not able to see exactly how, when we didn't observe the process that brought it into existence, we can still infer back into time and say there was an intelligent agent involved, because only intelligence is capable of producing information, and that's confirmed by everything we know about the way the world works.
Right. Now, what we're going to do, Stephen, we'll show the other animation, which is the sort of machines which are working within the mitochondria. I can't say I fully understand it, but I think it's similar to the previous one in that, you know, it is a manufacturing process, let's say. You know, and we call it ATP synthase. And as I started asking you before the program, we're not going to get to the depths of the process, but the animation is very clearly showing, you know, and illustrating what we're talking about. So I think it'd be nice for the viewers to see another animation. And this is called ATP synthase, and we'll discuss it later. ATP, by the way, I think stands for adenosine triphosphate.
Very good, very good.
So let's see how, you know, we create adenosine triphosphate from adenosine diphosphate.
It's been called one of the wonders of the molecular world, an amazing nanoscale machine. ATP synthase is a high-tech micromolecular power generator inside the cells of your body. It generates adenosine triphosphate, or ATP, an energy molecule that provides fuel that every one of your cells needs to function. Without this fuel, your cells will cease operation, and so will you. ATP synthase works like a rotary engine. The barrel-shaped rotator is composed of 10 to 15 protein parts called subunits. The rotator spins around, transmitting mechanical energy into the drive shaft of the machine, which helps make ATP. This drive shaft has a specially placed bump that opens and closes parts as the drive shaft spins around. This bump opens special protein subunits on the bottom of the machine. When the bottom subunits open, a spent energy molecule called adenosine diphosphate, or ADP, enters the machine. The mechanical motion causes the ADP to bind with an additional phosphate group, creating the ATP energy molecule. And the ATP drifts off into the cell, ready to power some biomechanical reaction. The ATP synthase machine has many parts we recognize from human-designed technology. A rotor, a stator, a drive shaft, and other basic components of a rotary engine. The ATP synthase is one of thousands of elegantly designed molecular machines inside your cells that make your life and all known life possible. ATP synthase. An example of intelligent design.
Thank you very much. I mean, that's another amazing animation, and had I seen that in my teenage years, I would have studied the subject, I think, Stephen. Absolute congratulations on at least creating a much broader popular appeal, I would have thought, for the subject. It's really brilliant.
One of the exciting things is that as you're able to see what's actually going on inside the cell, and I remember when I was first taking courses in molecular biology, that many times we would be learning about systems like the gene expression system that we saw first, or some of these molecular machines, and there would just be a sense of awe in the classroom, or students saying things like, "No way," that can't be, it's just, and so we had this, back to these guys in the 19th century. I love that quote from Huxley: "The cell is a simple homogeneous globule of undifferentiated protoplasm." He was one of the great scientists of the 19th century, and he was just fantastically wrong.
Yeah.
And people had no idea what even simple cellular life requires, let alone the trillions of cells that are involved in our bodies.
And this is essential, isn't it? The ATP synthase machine creates energy for the cell.
Right. It's the battery pack, if you will, of all cellular metabolism. All cellular reactions require the energy from this molecule ATP, and ATP is constructed in this very, as you put it, automated and complex process. And so we have in our bodies, I think there's something like a trillion cells in the human body, and every cell has mitochondria. In the mitochondria, this process is at work. It's going on right now in your cells and mine as we're speaking.
Right, and it's going on in the cells of our viewers. And as I invited you earlier, please do email and text. We want to get as much information from Seattle, Washington as we can in this program, but please do email in, text in on what you have seen, and we might be able to bring your questions into the last 10 minutes of the show. But Stephen, why this idea of a machine with a camshaft, as it were, that's clicking? It looks as, when we look at it, as though it's always been like that. What's the argument against it somehow just miraculously always having been in existence?
Oh, well, you mean, what is the...
Well, the animation is showing that something that looks as though it's just in perpetual motion. I suppose there's the macro argument that energy is being depleted in the universe, so it can't go on forever. But
well, right. I mean, we know these systems had to arise at some place. And so the question about biological origins has been, I think, brought into sharper focus because of the discoveries of the last 30 or 40 years of the information-bearing properties of cells and of the nanomachinery that is everywhere at work in cells. and which is completely necessary for cells to maintain their to stay alive. And so one of my graduate supervisors at Cambridge, who was also interested in the origin of life question, said that if we want to explain the origin of life, we have to that begs a prior question, and that is, what is it that we're trying to explain the origin of? And as we've learned, that life in even its simplest forms is immensely complex. It has an informational complexity. There are information processing systems, and living forms, even simple cells, are full of these nano machines. That has made the challenge of explaining the origin of life that much more difficult, because we don't know of any undirected processes that produce systems like that rely on digital code or nanomachinery. We only know of one cause that's capable of producing these types of effects, and that is intelligence.
Yeah. Now, we were looking earlier at the DNA. Apparently, the human genome has 3 billion characters in it. I suppose the question arises again, how on earth all that complexity can be explained other than a design. I know you're reluctant to talk about what other people postulate, but it just seems to me there's a big gap, in what I read in terms of how this whole lot came into being.
Well, right. I'm not reluctant about it. There's so many ways we could go because there's so many different theories. Right. But the basic theory, if you're talking about human DNA, would be to rely on the standard mechanism of natural selection acting on random mutations. And in the book we talked about in the last interview that called Darwin's Doubt, I addressed the problem, the many problems associated with the mutation selection mechanism. Many evolutionary biologists are themselves now acknowledging that the mutation and selection mechanism lacks the creative power to produce the fundamentally new innovations in the history of life that we see documented in the fossil record. I focused in that book on the problem of the very first animal life, but to build animals you need proteins, and to build proteins you need lots of new DNA. And just a couple simple analogies that we could get across in this context, I think help explain the problem. If you've got a section of functional computer code and you want to alter that to perform a new function, so you've got one program and you want it to become a new program, what's going to happen if you start randomly changing the zeros and ones? Are you going to produce a fundamentally new operating system or computer program, or are you going to degrade the information that's already there? The answer is obvious. You're overwhelmingly more likely to degrade the information that's there in the first program before you ever come up with anything remotely useful, i.e. another program or operating system. And that's the kind of problem that neo-Darwinism confronts. The mechanism can account for small changes, minor modifications, but if you start making multiple changes to those A's, T's, C's, and G's all precisely lined up, you're going to degrade the information that you have long before you get anything new and useful. So, and that's just one of, I look at 5 different separable problems associated with relying on mutation and selection to generate new information, the information necessary to build a new form of animal life, or to build us, for that matter.
I'm going to give you a quote, which I picked up. I can't even verify who it's from. It was just a quote that I saw this evening on the internet. But it just says the evolution, so there's a presupposition there, of ATP synthase, which is what we've just been viewing, is thought to be an example of modular evolution during which 2 functionally independent subunits became associated and gained new functionality. This association appears to have occurred early in evolutionary history because essentially the same structure and activity of ATP synthase enzymes are present in all kingdoms of life. How would you answer or respond to that?
Well, the second part of that statement is a fairly common kind of Darwinian reasoning. If we see similarity, then it must be because of common ancestry, because all forms of life have the ATP synthase. Then the ancestor of that form must have gone very, very far back. The problem with that, though, is that it presupposes an adequate mechanism that could generate that complex form of machinery in the first place. And as I mentioned, there's a tremendous problem accounting even for the origin of new proteins, even individual proteins, by relying on the neo-Darwinian mechanism of mutation and natural selection. Beyond that, these systems, the ATP synthase, the bacterial flagellar motor, and many other forms of nanomachinery that we find in cells often require numerous proteins working in close coordination before there is a selectable function. And so the kind of Darwinian just so stories about, well, this little part combined with this little part, and that built up to make a much more complicated machine, they may be superficially plausible, but they end up breaking down when you get into the details, because it turns out that there are lacuna or gaps in those causal stories, because, for example, another famous bacterial molecular machine is this bacterial flagellar motor that Michael Behe made famous in his book Darwin's Black Box. And people have tried the same strategy there. They've said, well, you've got this 30-part molecular machine, and it was built up gradually, starting from a subunit of that machine that has 10 or 12 parts. The subunit does perform a function, but it turns out that all of the intermediate stages between the 12-part machine and the 30-part machine perform no additional function, and there's nothing there to select. And so you have that kind of problem recurring over and over again, where natural selection selects for functional advantage, but the intermediate stages along the way lack functional advantage, so there's nothing really there to select, and you require these big jumps in complexity without a mechanism to accomplish that. And as I've argued in Darwin's Doubt and Signature in the Cell, just generating new proteins, just generating the information necessary to produce a novel protein lies beyond the causal power of the mutation selection mechanism. The problem is just too much new information is required.
Just for one.
Just for one.
Yeah.
And I go into the math of that in some detail in both books, but I also try to explain it with some accessible analogies. And I think the one I used a minute ago about just the computer code gets that across. What we know of systems that rely on functional information in digital or alphabetic form is highly specific in its arrangement. You start changing the arrangements, changing the letters at the given sites, and you're going to degrade the information long before you generate anything functional or useful.
Right, wonderful. Now, let's just remind the viewers that we're discussing Signature in the Cell, a book written by Dr. Stephen Meyer, available, Stephen, on Amazon. Is that the easiest way to describe it?
Amazon is, and the other online sellers, Barnes & Noble, the usual suspects.
Okay, brilliant. So maybe Waterstones, WH Smith, try them. Try their, computer database to see if they have it available. We have a number of emails that have come in. I might read a couple, without burdening you with all of them, Stephen, but some interesting ones coming in. Please comment on the supposed similarity between human and chimp monkey DNA and the argument that this is evidence for evolution. And I suppose coupled with that, by the way was from Ian, coupled with that from Jim, are the cells in animals less complicated? 2 emails.
The second question is real quick answer. Cells in animals are more complicated than the cells, than one-celled organisms. There are 2 types, basic types, eukaryotic cells and prokaryotes. Prokaryotes don't have a nucleus. The eukaryotes do and are much more complex. But the prokaryotic cells are mightily complex in their own way. So
but this might be shorthand for asking, are the cells in animals less complicated than cells in humans?
Oh,
essentially not.
I'd say they're on the same order of complexity. Actually,
yeah.
we have cells that are very similar to those of other animals. The first question is really a fascinating question.
Yes.
There's a fascinating story associated with that. We've all heard this argument that the chimp DNA and human DNA is 98 or 99% similar, and therefore that's strong evidence for the common ancestry of chimps and humans. The story has gotten quite a bit more complicated in recent years, however, as the chimp genome has been sequenced. That measure of complexity, or that measure of similarity, has been gradually dropping. But more importantly, the realization in recent years that human DNA and chimp DNA contains, let me back up. It's been long thought that most human DNA, most chimp DNA was so-called junk DNA, DNA that did not code for proteins, but was actually the detritus of the random trial and error process of mutation acting on, or natural selection acting on random mutations. So of those 3 billion human base pairs, 3 billion DNA characters, for a long time, people thought only about 3 million were functional. But in recent years, and there was quite an astonishing raft of papers published last fall by something here in the States called the ENCODE Project, documenting that at least 80%, and that's probably a low-end number, at least 80% of the genome is functional, including vast stretches of the DNA that were thought to be junk. That has the recognition that most of the DNA is functional, and there's very little junk DNA, if any, is causing a radical reassessment of that human-chimp similarity argument.
Now, the other one, just adding to this question a little bit more weight, that one of the strong arguments is that there's a sort of aberration or error which is common to both. And they say, well, that is more evidence that they could be a common ancestor.
Yeah, well, okay, well, let me get to that as well.
Yeah.
The key point, though, is that the chimp and human DNA, the assessment of similarity was based on comparison of the protein coding regions of the genome. 99, 98% similar in the 2 or 3% of the genome that are coding for proteins. We now know that the rest of the DNA is importantly functional. Overall, it's functioning much like an operating system in a computer, which is telling the data modules, in this case the protein coding modules, when to turn on, when to turn off. It's regulating the timing and the expression of that other information. Now, here's the key thing. In the case of chimp and human DNA, the operating systems, the non-coding regions that we now know to be importantly functional in regulating the overall expression of genetic information, is species-specific. It's importantly different. It's dramatically different in the two organisms. So if similarity of sequence was evidence of common ancestry, gross disparity of sequence may well be evidence of just the opposite, that is, separate origins.
So what about this similarity of the error? Now, I'm only saying this because this is what comes through our media, and all of what you've just said does not come through the media.
Right,
so
exactly.
I want to address that point.
Is very cutting edge, but this argument was made very prominently and famously by Francis Collins in his 2006 book, The Language of God, that you have these different types of genes, pseudogenes, that are clearly non-functional, and you find the same types in chimps and humans. Well, a lot of those judgments about non-functionality have had to be revised in light of the discovery of the junk isn't junk. And so those arguments are predicated on judgments of non-functionality. And what we're finding is that in one case after another, those claims of non-functionality have had to be reversed. So the factual predicate or basis of that argument is no longer in place.
Okay. Steve writes in, aren't simple arguments of left-handed amino acids, proteins, and right-hand sugars, DNA, and the chance of their makeup impossible when they all have to be either left or right and in the correct order purely on odds?
Yes, that's a wonderful question. The technical term for that is called homochirality. All the amino acids have the same orientation. Turns out that every amino, and I've got a visual aid if you'll permit me. I hope you can see this. Proteins are long chain-like molecules, and they're made of individual amino acids. And if the amino acids link up in the right sequence, the proteins fold into nice three-dimensional structures and can perform important biological functions. But it turns out that every one of the 20 protein-forming amino acids comes in two mirror image chemical forms, a left-handed version and a right-handed version. And to make a protein, you can only have left-handed amino acids. To make sugars, you can only have the right-handed versions of the sugar molecules. So you have this strange problem in biochemistry where there's this kind of a selectivity that's required. And it does, as your questioner alluded to, make the probability of generating even a single protein immeasurably small. And I discussed this at Signature in the Cell and look at actually a number of different what are called probabilistic hurdles in the field. You've got to get the right kind of linkage when you make proteins. It's something called a peptide bond, and the ratio of peptide to non-peptide bonds is about 50-50. You've got to get the right-handedness. You've got to get a left-handed amino acid, not a right-handed amino acid, and that's about a 1 in 2 chance, 50-50 probability for each site. And then you've got to get all of the amino acids to line up correctly. And when you take all those improbable conditions into account, that you get these extremely small probabilities associated with the chance origin of even a single protein. And the probabilities are so small that if you take into account every event that has occurred in the history of life, actually in the history of the universe, where an event is defined as an interaction between elementary particles, you still have an immensely small probability. The odds of building even a single protein are 1 in a trillion or less. So it's this is why researchers have given up on the chance-based theories for the origin of life, because chance alone doesn't account for the information to build even a single protein.
Now, Stephen, I just want to say thank you very much to everyone who's been emailing in. We're in the last couple of minutes of the programme. I'm going to risk asking, as you're a philosopher of science, this one from Andrew. We may not get through it all tonight, which will give us an excuse to invite you again, Stephen. But Andrew says, hi guys, you keep trying to discuss that evolution is not true, but you still fail to give an alternative explanation and prove that a god or a designer exists. DNA is a self-replicating molecule. That is the problem. If a god designed the flagellum motor, then he designed the machine that enables the cholera bacterium to swim through the human body system, causing 120,000 deaths per year. You've got a minute on that one.
One of the aspects of the Intelligent Design Research Program is to discern the evidence for aboriginal design and to distinguish it from subsequent decay. Not all ID advocates are Christians. I happen to be a Christian, and as a result of that, because of my biblical worldview, I expect to see not only evidence of original design, but also of subsequent decay. You see scriptural references to that, for example, in the book of Romans, that the creation itself is groaning for its redemption. I see the mutational processes that degrade information as responsible for that kind of decay. And it is interesting, in the field of virulent studies in microbiology, these really nasty bugs that are so harmful to humans invariably are the result of loss of original information. So that's the kind of thing that I, as a Christian ID theorist, would expect to see. Evidence of design, but evidence of subsequent decay of that original design. Just as in an automobile, you can see design in the automobile, but you can also detect the subsequent decay in the rust on the chassis, for example.
Okay, Stephen, thank you very much. That's a great answer. Unfortunately, we're now into our last few seconds, so I have to say thank you very much to everyone who's emailed. Sorry we haven't been able to read them all. Thank you again to Stephen Meyer in Seattle. Look forward to seeing you again. And Gordon has written Job 38, which I think we'll leave to another programme. But thank you very much, everyone who's joined us.
Thank you. Thanks for all the thoughtful questions, Pro and Colin. Very good.
Thank you very much, Steve.